And I'm a first-year PhD student at UC Irvine, and I'm in the Martin Lab. This is a map of California, and I just wanted to point out exactly where UC Irvine was, and that it is not the same thing as UCLA, which I think a lot of people thought after I had introduced myself, but it is actually located an hour south from UCLA and an hour north from UC San Diego. So in a way, I believe I live in the best place on earth, and I have the best of both worlds if I wanted to travel to one of those cities. Also, before I start my presentation, I wanted to point out that I'm not a physicist or a mathematician, but I'm a chemist who studies biological structures, and therefore structure or equations like these will not appear in my presentation, and I still have no idea what they mean. So anyways, but structures like these, which are represented here by the colorful pictures, will appear in my presentation. And here we have the DNA molecule, and on the left side is RNA folded up into this structure, and here are the proteins, which are actually membrane proteins here. So since a lot of you guys are physicists, I'll just go over some of the key concepts of each of the macromolecules which I just showed. Deoxyribonucleic acid, or DNA for short, is found in either A, B, or Z double-stranded helix in the nucleus of the cells, and it contains genetic information. And DNA can be transcribed into ribonucleic acid, or RNA, which is single-stranded and is folded up into these structures called aptamers and ribozymes. And what aptamers are so they're sequences of RNA that bind to other target molecules in order to then regulate the RNA's activity. And ribozymes are actually enzymes which catalyze chemical reactions. And another function of RNA is to express genes. And RNA can be translated into proteins, which are chains of amino acids that have many functions to them. And what our group actually is more interested in are membrane proteins, which have also many functions, but some of those functions are cell communication and the transport of nutrients across the cell membrane, which are important for homeostasis. And because all of these molecules are crucial for living systems, it is highly important for us to understand their structures. For instance, some pharmaceuticals are interested in understanding membrane proteins, which act as receptors in order for them to synthesize new drugs for those receptors to see how the cell responds to those new drugs. DMPC, DHPC mixtures are a mixture of these long chain and short chain phospholipids, which exhibit interesting liquid crystalline phase behavior. And what happens is actually at low temperature, these mixtures are liquid and in an isotropic phase. And they have these sort of disc-like micelle structures to them known as bicelles. And at a higher temperature, the mixtures are actually gel-like and very viscous, and they're in the chiral pneumatic phase, which is able to align in the magnetic field. And instead of being disc-like, they're these worm-like micelle structures. And so DMPC, because they're able to align in the magnetic field, these mixtures have been used as aligning media for other biomolecules which do not have liquid crystalline properties and therefore are not able to align in the magnetic field. And also it has been shown that divalent cations and cholesterol sulfate can help the alignment of the bicelles. So the way I study the alignment of the mixtures and of the biomolecules is using nuclear magnetic resonance. And this technique is highly used by chemists for determining structures of molecules. And it's a very complicated technique that I'm still trying to understand, but the very basics of it revolve around nuclear spins and their electrons. And so most of the nuclei that the organic chemists are interested in have a spin of plus or minus 1/2. But I'm also interested in deuterium, which has a spin of plus or minus 1. And so in the presence of an external magnetic field, the nucleus and its electrons precess around that magnetic field in the direction of the magnetic field at a certain frequency. And then when you apply a pulse of radio waves at another frequency, it then excites all those electrons to a higher transition state, so a higher energy level. And they then begin to possess opposing that magnetic field. And what we observe is actually the relaxation of those electrons from the higher energy level to the lower energy. And we see that in the spectra after it's Fourier transformed. So it's pretty tough stuff. So I use that NMR technique to then study structures of other biomolecules in the aligning media of those bicelles that I had just described. And currently, I'm actually interested in the structure of this Dickerson dodecamer, which we all heard about from Noel Clarke's talk. And what's cool about it is that it has this palindromic sequence, so determining, we only need to determine half of its structure to know the full structure. And even though Noel did talk about that it has liquid crystalline properties at certain concentrations, actually did not take that into account. So don't think of this DNA as being a liquid crystal. Think more about the bicelles as being the liquid crystal and orienting that DNA molecule in the magnetic field. And besides studying the structure of the biomolecule, I also want to study the alignment of the bicelles with this cholesterol sulfate at the lower temperatures, as well as study the phase transitions at the lower temperatures. So here we have my deuterium studies of the lipids in the absence and presence of DNA over the 5 to 35 degree temperature range. I used a 600 megahertz magnet, and what happens to my sample is I put it into a NMR tube. So instead of having it in between plates, as you all I think are more familiar with, this is actually like in this long, not capillary tube, but just like a thin tube. And so this Q right here represents the long chain to short chain ratio. And what's interesting about this to note from these spectra is that, so at 5 and 10 degrees, the single peak corresponds to the isotropic phase. And then as we increase the temperature at 15 degrees, there's a quadrupole splitting happening, which corresponds to the anisotropic phase. And then again, this splitting diminishes for the most part here, but to a single peak, isotropic again. And then at the higher 30 to 35 degrees, again, we see the anisotropic, which is interesting. So there's 2 phase transitions going on here, which we don't know what they are, but there's something happening. And then also the phosphorus studies, which I did also support my deuterium data. So here we have one peak, and then this is the peak I'm looking at. And then at the 15 degrees, we get this new peak appearing corresponding to the anisotropic phase. And then at the 30 and 35 degrees, that peak begins to grow even more. So that's that. And then moving away from looking at the phase transitions, we're now going to look at how the lipids, what effect the lipids have on the DNA in terms of the alignment. So these are just proton NMR spectra in the nucleotide region. And so here is just DNA without the lipids, and here I added the lipids over the 5 to 35 degree temperature range. And what happens is we see more peaks begin to appear, so something is going on there. And then at the higher temperature, those peaks begin to broaden out and decrease, which most likely corresponds to the fact that it's becoming very gel-like and the magnet is kind of freaking out. And so the main splittings which I'm interested in are from these cytidine protons, because they can couple to each other, and therefore I'm able to see doublet peaks, such as these right here. I don't know if you can tell. So what I want to see is if the DNA is able to be weakly aligned by the lipids, I should see a change in those splittings. And that change in the splittings is going to correspond to a residual dipolar coupling. So then from that, I can calculate nuclear distances in atoms of the structures. If it's, I can do either short range, or if I do perform 2D studies of nosy experiments, which are long range protons, I can then calculate the distances between the long range protons, which is cool. So this is what a 2D spectra looks like, and I'm going to skip it because I don't have, nothing too interesting. And so for my short results summary, I see these two-phase transitions happening with the deuterium and the phosphorus. I'm not too sure exactly what those are yet. And then we also see the differences in the 1D spectra in the presence and absence of DNA. And so there's something going on there as well. And I have a lot of future goals, actually. So I want to change the concentration of the lipids and the cholesterol sulfate in order to determine what that has on the phase behavior of the lipids, as well as add divalent cations, which have been shown to do something to the alignment, and then also use microscopy and light scattering techniques to determine what are those phases that I'm seeing. And then, because NMR doesn't really tell us too much, right? And so then I also want to perform more 2D experiments in order to then calculate those residual dipolar couplings to then get my nuclear distances, and then use other lipid systems which actually work better at the lower temperatures. And then perform similar studies with RNA, because that's the more interesting molecule because of all the structures it forms. And then, so acknowledgements, I want to acknowledge Andrei Luptek, which is one of our collaborators in the pharmaceutical sciences department at UC Irvine, and he's been awesome with helping with figuring out how to use the NMR as well as working with the biomolecules. And then Rachel Martin is my advisor. I'd like to thank her, and then Amanda Brindley, who actually did all the cholesterol sulfate studies and the divalent cation studies with the system. And she's one of the students from the lab that graduated last year. And then Phil Dennison, who is the NMR facilities director. He's been great. And then also, of course, my lab group, and then NSF for funding the project, and then obviously iCamp 2013 for giving me this awesome opportunity to present and be a participant here. And I had an awesome clicker question, but yeah, I had to put a clicker question into this lecture, you know. So I was going to have you guys pick what I actually like for lunch versus what you guys like. So yeah, the end.
One quick question.
But you answered the question.
Right, the back.
We can't. There's no clicker.
Right at the back.
So didn't you observe your sample under the microscope yet?
So that's my next step, is figuring out exactly how to get the conditions right to do that. Because you have to look at it over the temperature range, and then you want to, because the way, I'm not sure what the best way to do it, like put it on the slide, or like does anyone have suggestions? Like what do people use for capillary tubes? I'm not sure.
Normal sandwich cell?
Sandwich cells?
Yeah.
Yeah, that's my next step. So.
Okay, thank you very much again, and it's time for coffee.